Thank you very much. Well, thank you very much, Astrid. Good morning, everyone. The experimental part, which I'm going to present today, was based on my lecture, which I gave in 2008 in Stockholm. Because at that stage, I felt that we were a little bit tired of talking for 3 decades on papillomaviruses and other agents in human cancer. And we thought that some novel approaches were required to support the search. So I presented a lecture, The Search for Human Tumor Viruses and Infectious Agents in Human Cancer, Where and Why. And in part, based on our studies which we did previously on single-stranded DNA viruses, we developed a specific approach on which I'm going to talk today, particularly in view of the fact that although epidemiological evidence existed for quite a number of common cancers that may linked to infectious events, and in spite of the fact that deep sequencing of those types of tumors didn't reveal any convincing data on the role of infectious agents, we decided to look differently into this whole story. Now, I've shown this picture at many other occasions. The basic idea originated from the fact that we know in humans a number of infectious agents which are persistent infections, latent infections for lifetime in part, and do never seem to convert to induce malignant growth. On the other hand, if those agents, some of them, are injected into laboratory animals, they are in part highly tumorigenic. And this was under conditions where they cannot reproduce, whereas in the productive system, the humans in this case, they were apparently non-oncogenic, but under conditions of non-reproductivity, they became oncogenic. This triggered the idea, is it possible that in the animals with which we live in our close proximity, farm animals or pets, that they may harbor similar agents which in the native species in which they replicate are not tumorigenic at all, remain in part in them for lifetime and being excreted in several of their fluids or materials, but upon transmission to a species in which they cannot replicate, in this case to humans, that they may become tumorigenic. So that triggered the idea, and we basically started to work initially on colon cancer. Why colon cancer? Because there were a large number of reports on the consumption of red and processed meat as risk factors for the development of colon cancer, and indeed in countries with a very high rate of these types of cancers, of red meat consumption, there was also a high rate of red meat consumption as well. So we suspected that this may be some kind of a specific event, although since 1978 approximately it was suspected that the broiling, frying or barbecuing of meat with the production, as Sugimura showed it initially in Tokyo, that chemical carcinogens arise under those conditions, in particular aromatic hydrocarbons, nitrosamines and a few others, that they might be the reason for this increased risk. Yet the same chemical carcinogens arise if you broil, barbecue, or fry chicken or fish, as Sugimura already noted in his early studies. And yet, even in populations which are mainly consuming those types of nutritional things, there was not an increased risk in fish consumption, even a reduced risk for colon cancer. And also the epidemiology, the geographic epidemiology, suggested that there were some discrepancies because Mongolia is a country with a high rate of red meat consumption and a very low rate of colon cancer. And so this suggested to us there must be something else behind this whole story. And by studying the literature, it turned up that a large number of different types of cancers have been reported to be linked either to red meat or dairy product consumption. And the list is very long. It's longer than shown here on the slide. particular colon cancer, even breast cancer, lung cancer as well, besides smoking, of course, as a major risk factor. Prostate cancer have been linked, and the other ones with a relatively low consistency, but I may come back to the one or the other later on. So we developed a kind of a working hypothesis on this type of speculation, which is outlined here. The lines here should represent the lifespan, let's say, of about 80 years, and during this period of time, the consumption of raw and undercooked red meat may lead to the uptake of some suspected putative carcinogenic agents, which turn into a state of latency in specific types of cells, in this case in the cells of the colon tissue, persists there, probably for lifetime, and only under specific conditions by interaction with some chemical, physical, or other types of mutagenic or epigenetic modifications inducing compounds, they may lead to some type of cancer. I should mention here that there's not a single agent which is linked today to cancer among the 21% which have been linked to human cancer globally, which by itself is sufficient for cancer induction. They all require some additional modifications of host cell, of the respective host cell DNA, either by mutational or epigenetic events. So we took this also for granted for this type of system and suspected as well that inherited genetic modifications would play a similar role than acquired modifications in this respect by leading to a synergistic effect with those types, but enhancing the risk for the development of the respective form of cancer. Well, because there was some kind of there seemed to be some kind of geographic specificity of this type of cancer in relation to dairy cattle farming and consumption of red meat from dairy cattle. It seemed to be a species-specific effect. We started out with some experiments to look for some possible agents which may play a significant role in this case, and this was based in part on the fact that we had studied before single-stranded DNA agents in humans, the so-called TT viruses, which are widely spread in human populations, where we failed to find any evidence for a link to human diseases. Now, so we started to isolate from 130 cows, serum, which we received from the veterinary station in Leipzig, and also the dairy products, which we all bought around from the supermarkets around Heidelberg, and analyzed them for some putative agents which came into consideration. In fact, the result was a bit surprising, because we came up to today to more than 40 individual different types of single-stranded DNA, of small size, circular DNA, of small sizes of about 1,200 nucleotides up to 3,000 nucleotides, which, as I could state already at this stage, to our knowledge, never have been seen integrated into host cell DNA, but persisted obviously episomally. One of the striking features here, and also the suspicious one from the beginning, were that of the 4 groups which we isolated, listed here, that 3 of them showed a very strong relationship to bacterial plasmids, which of course suggested instantly that they might represent contaminants and not something real. But it was interesting, I will come to this in a moment, that at least 4 of the isolates originated from patients with multiple sclerosis. 2 of them directly from autopsy brain tissue of such patients, 2 of them from the serum of such patients. So this was, at least to some degree, suspicious for us. And there was another type of cancer which came into the fore in the course of these studies, namely human breast cancer. Human breast cancer, because there's a remarkable, if you look into the geographic prevalence of colon cancer, as shown here by the dark areas in the lower part of this picture, some remarkable similarity to the geographic epidemiology of human breast cancer, in that countries with a high incidence of colon cancer usually also reveal, like Argentina, Australia, and so on, a relatively high incidence of breast cancer as well. Now, since we know that there exists a mouse model for memory cancer, which is caused by an agent, the mouse memory tumor virus, of course it was interesting for us to see whether this agent in mice is transmitted by the milk of the feeding mothers to the newborn babies and settles in the intestinal cells, or particularly the pious patches, infects T and B lymphocytes, induces what is called a super antigen. They swell, virtually produce large quantities of virus particles, and the viral particle dose appears to be responsible after elution into the bloodstream, that they reach the mammary gland and induce cancer there, particularly under hormonal stimulation, because they show promoters which are highly hormone dependent in their reactivity. It's interesting to compare the pattern of breast and colon cancer on a time scale in different parts of the world, particularly for colon cancer. It was evident in Japan and Korea that about 15, 20 years after the Second World War in Japan, the colon cancer rate was rapidly rising, as it did about 15 or 20 years after the Korean War in Korea, whereas India, which of course is one country where cattle products, at least cattle meat, is not being consumed at all by the Hindu population, showed a low incidence of colon cancer. In breast cancer, the situation is quite similar, but not identical. And that was interesting because, in particular, in India, the scales here are different on the two parts of the slide. In India, the breast cancer rate increased, particularly during the past 20 years, quite substantially. And India had introduced big farms of dairy cattle around 1980, maybe a little bit earlier even, in order to improve the nutrition of the young babies. And it's by many of the pediatricians that's recommended to use cow milk relatively early. In Japan and Korea, the rates increased also, but not at the same rapid pace as did breast cancer. So we thought, since the low increase of breast cancer in Japan and Korea contrasts the steep increase of colon cancer, that probably there's one difference between breast cancer and colon cancer. In colon cancer, if there are infectious agents involved, they are directly reached by the nutritional aspects in the intestines, in the colon specifically. But for breast cancer, there's some kind of transport necessary from the intestines to the breast tissue. And this may occur systemically, or it may occur by the transmission of specific cells. But it would indicate that somehow the agents, if they are responsible for these types of cancers, would differ from each other, but they still could originate from the same species. So we modified the hypothesis for breast cancer slightly by assuming that in particular in the very early period of life, namely in the first, probably mainly in the first, but maybe in the first 2 years, that's the most important and critical period for taking up this putative agent, which may later on lead to breast cancer, that it leads to either by systemic or by a transported infection to the transport to the breast tissue, latent state, and that synergistic interactions by either acquired genetic modification, similar to what I pointed out for colon cancer, or by inherited genetic modifications, would interact finally to lead to precursor lesions and to the respective form of cancer. So if milk contains the putative infectious factor, it has to be transported to the target cells in the breast, and this is most likely to evoke an immune response, which probably subsequently, in subsequent infections, would neutralize the infectious agents, so that we suspected that this period is really the critical for events which take place 40 or 50 years later, or even sometimes earlier and sometimes later, which is quite analogous to some of the tumor viruses which we know right now, like hepatitis viruses, also parasitic infections and bacterial infections, Helicobacter pylori, which usually require something like 40 to 60 years of infection prior to the development of the respective types of tumors. It's an interesting study which was published in Sweden on lactose intolerance, and the uptake of dairy products. Of course, lactose-intolerant patients or persons do not take up or try to avoid as much as they can dairy products because they cannot tolerate them. And in this study, the authors took advantage of the Swedish Cancer Registry, identified about 23,000 persons with lactose intolerance, and compared them to about 70,000 siblings and parents, optimal controls, which were lactose tolerant. And the interesting result here was that among the lactose intolerant persons, there was a reduction in the breast cancer risk by 21%, and what is even more striking and interesting, a reduction in lung cancer risk by 45%. I didn't find any data on the smoking habits of lactose-intolerant persons, but it seems unlikely, really, that this is the sole factor here in this drastic reduction of lung cancer incidence. Now, do we acquire infections from bovine milk consumption? Well, as I said before, epidemiological data suggests that early events should play a role for the risk of some cancers and other chronic diseases. There's an interesting aspect coming up here, namely that prolonged duration of breastfeeding turns out to be an important protective factor. That has been investigated in recent years in some more detail. If you take this as a period after delivery of breastfeeding, let's say for 6 months or more, then within this period of time, the babies take up some very specific sugars in the human milk, like 2' fucosylactose and 3' fucosylactose, lactose tetraose, and also to some degree interferon gamma. And at least the sugars here, they are quite specific for human milk. In cow milk, they are not present. There are other sugars present, and these other sugars in cow milk have a protective effect for infections which occur in young cattle, in newborn cattle babies. Now, obviously, this provides some kind of protection. I will come to this in a moment against specific types of infection, because these sugars determine the receptor properties of specific receptors in the glycoproteins of the cell surfaces. During the weaning period, subsequently, and the uptake of dairy products, there's one other important event taking place, namely due to the presence of specific sialinic acid, namely N-glycolylneurominic acid, for instance, which is not present in humans and cannot be produced in humans. These new types of sialinic acid become incorporated again into surface glycoproteins and act as novel receptors for specific virus infections. These are some rather remarkable and interesting events which have been studied in a number of laboratories during the past couple of years. And we assume that in this period, particularly during a slow weaning period, that there is due to the maturation of the immune system, a protection taking place due to neutralizing antibodies. This is a little bit highlighted here. The sugars in human milk block specific virus receptors and prevent infections, for instance, of noroviruses and rotaviruses. And these are infections which, at least in very young babies, are deadly infections and are the cause for death in some of the developing countries to a very large degree. The uptake of these new type of neurominic sialinic acids, the NOI5GC as it is conveniently abbreviated, during the weaning period by cow milk and meat products results in their incorporation into cell surface glycoproteins and forming novel receptors for specific infections like the human polyomavirus type 9, which could not infect at an earlier period, specific parvo, even specific strains of influenza A and paramyxoviruses, and probably also for additional agents. Interestingly, these novel receptors are immunogenic, and they may cause inflammatory reactions at the same time at certain sites. Well, it's interesting to note that there have been a number of reports on the protective effect of prolonged breastfeeding for more than 6 months and more on chronic diseases, particularly for the acute lymphoblastic leukemia, also for Hodgkin's disease, and interestingly too, for multiple sclerosis. There were less consistent reports on neuroblastomas, even premenopausal breast cancer, but it's of course very difficult in cancers which arise after 30 or 40 years or so to date this back to some events during the baby period, and even diabetes type 1 and 2. Now, let me come back to the isolates which we obtained, and particularly the isolates which were obtained from multiple sclerosis directly from lesions or from the serum of multiple sclerosis patients. Of course, as I said before, some of them, particularly this group, group 2 and group 4, they showed a remarkable similarity to bacterial plasmids. Of course, it raised the suspicion, are they contaminations? In fact, so it was the first step which we did initially to look whether transfection into various types of cells lead to an expression of those genes. Indeed, it does. All those which have been tested so far of the types which we have isolated produce messenger RNA in human cells, and they also produce proteins under those conditions, and these are the colleagues who did most of this work in my lab. It's interesting here to see that a couple of neurodegenerative diseases, highlighted here for multiple sclerosis, have been linked to milk, in this case to farming, vitamin D deficiency, herpes virus reactivation, and a couple of other factors. But meat and milk and farming seems to be a relatively consistent feature. Of course, for mad cow disease, it is well established that it is an infectious disease, but even for Parkinson's disease, amyotrophic lateral sclerosis, and to a low degree also for Alzheimer's disease, it has been reported. Now, for multiple sclerosis, there have been as main risk factors, 3 factors claimed to play a significant role: cow milk consumption in childhood, the vitamin D deficiency later in life, and reactivation of latent herpes virus infection, in particular Epstein-Barr virus, but also human herpes virus type 6 as main factors of reactivation. Now, this was quite interesting for us because we had studied some decades earlier, really, in Freiburg still, the fact of herpes viruses, also of adenoviruses, in cases of co-infection of cells which contain genomes of small DNA viruses. It's well known that adenoviruses, but also herpes viruses, amplify rapidly parvoviruses, the adeno-associated virus, for instance. But even in cells which we transfected at the same time with a TT virus, a human single-stranded agent, we could note that we found these clusters of particles which we could not specifically identify, but they're most likely particles formed by the TT virus transfection in cases where, and this is shown by these arrows, Epstein-Barr virus reactivation took place. We found it only in those types of cells, and we have noted at that stage that also polyoma viruses, other single-stranded agents, and also papilloma viruses can be amplified, usually by inhibiting the helper viruses, in this case the Epstein-Barr virus, but by promoting the replication of the smaller DNA, probably by competition for DNA analysis. So that was an interesting aspect. The other interesting aspect is the vitamin D deficiency, which has been frequently noted as a risk factor, and this serves as an explanation for the north-south gradient of multiple sclerosis in the regions of low sun exposure, where multiple sclerosis in the northern and southern hemisphere is relatively high. So again, during the Freiburg period, we have studied the role, in particular, one of my co-workers, Georg Bauer, the role of growth factor in Epstein-Barr virus induction. And this is TGF-beta, tumor growth factor beta. It has been identified at that stage. He called it initially the Epstein-Barr virus inducing factor, and later on it turned out to be identical to TGF-beta. That this factor is inducing Epstein-Barr virus, and according to the literature, those experiments we didn't do ourselves, it turned out that TGF-beta is regulated by vitamin D signaling cascades, triggered cascades, in the sense that vitamin D is suppressing TGF-beta production, and thus should lead to a reduction of reactivation events, and vice versa, vitamin D deficiency should lead to an increased induction. There are also some clinical data supporting this, because people working in the Antarctica, they have a very a high rate if they work for 6 or 8 months without sunlight exposure, high rate of Epstein-Barr excretion in the saliva, and vitamin D supplementation is blocking this effect. So we developed a model here for multiple sclerosis, which is schematically outlined here. There are 2 preconditions for the event. Namely, the first precondition would be a rare dual infection of individual cells within the brain by what we call the bovine meat and milk factor, and a latent infection by Epstein-Barr virus, or also human herpes virus type 6, but let's remain to the state with the Epstein-Barr virus model. The second precondition would be a vitamin D deficiency, the activation of TGF-beta and Epstein-Barr virus reactivation. So in those cells, upon reactivation of Epstein-Barr virus, the amplification of small genomes should take place, as it is shown, for instance, here in this picture, where in the center you see some Epstein-Barr virus particles and the amplified small other agents, which probably represent the T viruses, but they have not been specifically identified in this case, and that the excretion of those types, either of particles or proteins by exosomes or by infection of the neighboring cells, would then lead to the uptake of such materials by the neighboring cells. And since we would all anticipate, and we can prove it today, that everyone has antibodies to these types of bovine milk and meat factors, due to an existing immune response, we would anticipate a focal immune response, the destruction of cells which produce the respective proteins, and the onset of disease. This shows a lesion here of an early lesion of a multiple sclerosis patient, which I owe to the Department of Neuropathology of the University of San Francisco. And what we can observe ourselves, that in MS patients, although the vast majority of individuals, adult individuals, have antibodies to our factors here, that patients with multiple sclerosis have a very high titer, in average, about 5 to 7 times higher titer to this antigen than it's similar to Epstein-Barr virus situation in some respects in Burkitt's lymphomas and nasopharyngeal cancers. So in line with this model, we would also expect that there should be ABV signs for Epstein-Barr virus reactivation, which have been even described earlier by other authors, and the EBNA1 titers should be consistently elevated. They are consistently elevated in MS patients, as we could also show it in our own studies. So that seems to support this model. In a formal sense, it doesn't prove it really, but I think it's very much in line with what we expect. Interestingly, more than 50% of colon cancer biopsies and colon polyps contain Epstein-Barr virus DNA with inflammatory infiltrates, several of them with evidence for Epstein-Barr virus reactivation, and vitamin D deficiency in these patients with preneoplastic and neoplastic colon tumors has been reviewed recently, and there's even a publication by an Italian group showing a reactivation of cell of Epstein-Barr virus in the inflammatory infiltrates around colon polyps and colon cancers, and also the expression of latency Epstein-Barr virus RNA under those conditions in the inflammatory cells, again showing that there's a widespread presence of ABV in those cells as well. So what is the genetic activity of our isolates in human cells? Well, we see it right now that the circular single-stranded genomes persist generally as episomes without evidence for integration into host cell DNA. All those which have been tested up to now upon transfection are transcriptionally active in human cells, also depending on the genotype at significantly different levels. The BMMF group 1, and that's the only one which we have carefully investigated so far, shows in part substantial sequence homology to specific Acinetobacter baumannii plasmids, but we do find RNA transcripts that are terminally polyadenylated and also have evidence for splicing events occurring. So somehow these originally plasmid-derived sequences seem to have adapted to the proliferation in human cells. In some human cell lines, the transfection appears not to result in replication, but specifically in cells of the lymphoid lineage, DNA replication takes place. I'll show you an example in a minute. And abortive infection is completely of these small particles of circular molecules of about 1,400 nucleotides or so is complemented by co-transfection with larger genomes, those which have something like 2.7 or so nucleotides, 1,000 nucleotides. And as I said before, a high percentage reveals antibodies to the major protein. This shows in transfection of a specific cell line, in this case of a Hodgkin lymphoma-derived cell lines, which Konstantina Falida did in our lab. She's here among the audience, I presume. And there's good evidence that there's an increase of replicative activity, and we even see some kind of the formation, some of them have been sequenced, of defective, but probably defective interfering DNAs, which arise under these conditions of replication in those types of cells. The production of infectious particles is presently under investigation. We have some suggestive evidence, but this needs to be further elucidated, but it looks as if this is also the case. It's interesting here to note that infectious mononucleosis as an Epstein-Barr virus-linked disease has been repeatedly identified as a risk factor for the development of multiple sclerosis in the early period following Epstein-Barr virus infection, by the way, also Hodgkin's disease. And small 25 nanometer particles can be seen after Epstein-Barr virus reactivation in several Epstein-Barr virus transformed cell lines, particularly Burkitt's lymphoma lines, and also in EBV infection immunosuppressed individuals, in hairy leukoplakias, these white plaques on the lateral part of the tongue, where Epstein-Barr virus can be seen in large quantities, but similar particles were observed initially by Greenspan in San Francisco. And significantly elevated antibodies, I mentioned this before, have been demonstrated in MS patients. The latter increased during exacerbation. This shows this typical particle, in this case here, in a Burkitt lymphoma cell line, which has been induced to Epstein-Barr virus reactivation. And these particles, I noted it in several of the lines which existed in the laboratory, are being formed, and they are clearly different from the Epstein-Barr virus particles, which you can see here. And this is a slide which I owe to Greenspan from California, of a hairy leukoplakia, where you see here the typical Epstein-Barr virus particles, and again, it's a different magnification. I'll finish in a second. A different magnification of these small particles, and we suspect that this uptake of the sialinic acid, there are few pieces which suggest this very strongly, might be an essential component of the BMMF receptor as well. I conclude. So we isolated novel viral families of bovine meat and milk factors, and it identified several of these agents as zoonotic infections in humans. In spite of a substantial relationship to specific bacterial plasmids, we documented the adaptation to genetic activity and DNA replication in human cells. Antibody production in healthy humans demonstrates previous exposures to these infections, and epidemiological and our serological and molecular analysis link specific types of these infections to neurodegenerative diseases, here specifically to multiple sclerosis, and less clearly at the moment to some common human cancers. So epidemiological data, as I said before, suggests that they may play some role in other cancers. The novel aspect seems to be that these isolates represent an example of bacterially derived plasmids adapted to infection and replication in human cells. Interestingly, wherever deep sequencing has been done, those types of bacterial sequences are thrown out instantly and are not observed under those conditions, of course. For us, it seems to open a new research field in human chronic diseases at the interface between bacteriology and virology, cancer, and neurodegenerative. I'm finishing with two very quick more slides. Agrobacterium tumefaciens is a good example in plants where you get tumors induced by the transfer of a plasmid, of a bacterial plasmid to plants. This is a group which is quite engaged working on this type of subject, and as Michelle de Villiers is really quite substantially involved as well. She happens to be my wife as well. And these are our external collaborators at the University of Leipzig. Thank you very much.